So here are the three parts that we will be covering for the rest of this session. So first one would be the input, considered as the input, the sequences that you will have to submit or query. The second one would be the methods that you would apply to align your sequences, and then the expected output. So let's start with the input. So everything is rendered possible now in comparative genomics thanks to the availability of a huge number of genomes that are sequenced. And in fact, these numbers reaches thousands of genomes. They come from different kind of organisms, from different taxa, from different sizes, from the smallest to the biggest that you have here as an illustration. But these are changing rapidly. And as we saw it in the previous session, the development of the whole bunch of novel technologies, second and third generation technologies for sequencing, allow now even bigger genomes to be sequenced. So you have here an example of the genomes that are available through NCBI. And you see how many different genomes are deposited now for eukaryotes, prokaryotes, or viruses, etc. And these, there are different repositories that offer today the possibility to browse and retrieve information and exploit the data that is deposited in databases. So maybe two key journals to search for novelties, related to this query of databases could be found in nucleic acids research or database. I'm giving you here examples of these issues and examples of articles that have been published. Here is the one that is highlighting the use of assembly, which is a resource for assembled genomes at NCBI. And in fact, this database reports metadata such as the assembly, statistical reports, etc. And it's also tracking the relationship between the database consortium INSDC, if you remember, we saw it on the first session, and it analyzes the relationship and tracks it between INSDC and the NCBI RefSec. So what the user can do with this kind of repository or resource is query the assembly resource directly or by browsing in available assemblies for a particular organism. Another example is one of the tools that has been developed to highlight the possible exploitation of sequencing throughout the databases. And because we have now an increasing number of genomic sequences, we have databases or repositories or resources that are offering the possibility to develop distance determination based on whole genome information. And there is an estimation that is called ANI, which is the average nucleotide identity, that is calculated from pairwise comparisons of all sequences that are shared between two given strains. And this has been proposed as a new metrics for bacterial species in this publication. So there are different file formats that can be accepted. And I'm just highlighting here the FASTA format, for example. A FASTA format would basically start with a superior sign, then an accession number and characteristics like the name, the name of the gene. You can also copy and paste raw sequence like here, and you can also use the accession number to query the database. So you can use whole sequence or subsequence. So this is basically the web page for BLAST as an example. So we will be seeing how BLAST works later on. But I would just wanted to highlight one of the web resources that would allow you to query one sequence or BLAST exist even for genomes. So you have here an example of the web page where you put your sequence and for which you can query representative genomes or all genomes, choosing from the complete ones to draft versions of the genomes. Here is the example of the access to the microbial nucleotide blast. This is just an example. We will be highlighting these, the characteristics of the blast later on. So let's talk about the methods now. So in fact, DNA sequence alignment consists of the alignment of two DNA sequences in order to identify the regions that are showing sequence similarity. So this is a very basic definition, and this highlights the regions that are showing relationship, if you remember it, in terms of evolution, structure, and function. I'm really highlighting these three keywords throughout this session, because you will find them anywhere. So let's imagine that we have two basic simple sequences to compare. How we would perform a sequence alignment? Well, here we can do it even by hand. So aligning these two sequences would be basically finding the blocks of sequences that are exactly the same between the two sequences. So here we are finding three different blocks. Now we have two extra bases for which we do not have any correspondence, and for these we would be, in order to maintain the block alignment, we would be adding something that is called a gap. So how would you interpret this kind of alignment? So well, if you consider that these two sequences share a common ancestor, then you can imagine that in order for these two sequences to be aligned, yes, then we have to introduce single point mutations to these alignments, and a gap would represent an insertion or a deletion in a nucleotide, in one or both the sequences, of course. So, in fact, this would show how these two sequences, or two genomes, or two organisms, diverge from one another, and this is at the basis of the evolution theory between that we are searching for between two sequences. So, for the sequences that would not be as simple as these ones, like long sequences, for example, or more complex sequences that would be highly divergent and containing different kind of variation, or large number of sequences, we can obviously not do this by hand, and we would need algorithms, and this is why these have been developed. So the two key approaches for sequence alignment is global alignment or local alignment. I'm putting here two examples of sequences. So these are two sequences, and the same are here to highlight or to explain the difference between a global and a local alignment. In this case, we optimize the alignment in order to span the full length of the sequences that are aligned, as you can see it here. Whereas in the local alignment, we would optimize the alignment to take into account the regions of the highest similarity between divergent sequences, like in here, for example. So this only highlights, in fact, the possibility to use algorithms when you have more divergent sequences, so you would not be able to align them on the whole sequence, but instead you would be searching for regions of highest similarity. And these algorithms, efficiency and choice would depend on the number of sequences to compare. So two other key notions here would be the pairwise alignment compared to the multiple sequence alignment. So pairwise alignment is sequencing of the alignment of two sequences, with the output being correlation or knowledge concerning the function, the structure, and the evolutionary relationship between these two sequences. Whereas multiple alignment is basically sequencing 3 or more sequences, and the output would be the homology, the relationship of homology, and the evolutionary relationship. So let's start with a pairwise alignment, and we'll be seeing examples of pairwise alignment and examples of multiple sequence alignments. So a pairwise alignment is an optimized local or global alignment of 2 sequences. So there are 3 main methods, one that is called a dot plot or dot matrix, another one that is called dynamic programming, or the word-based method. So let's start with the dot plot method or dot matrix. So basically what you do in here is you take 2 sequences, A and B, as you can see them here. So this would be your query, and this would be your reference. And what you do is you align these 2 sequences based on a 2-dimensional matrix. Each time you would find an identity, so you use kind of windows, and each time you find an identity, then you add a dot. So the diagonal, aligning all over the diagonal, would show a highest similarity. So you have here examples of a dot plot of one sequence compared to another, and you have examples of interpretation that we can make. So if here you had a reference with AB and your query contained an internal sequence, then this would be represented this way with a dot plot, and this would indicate an insertion into this query. The inversion would be represented this way, and you have a more complex rearrangement into the query that is highlighted here. So dynamic programming is another kind of pairwise alignment approach. It uses a scoring matrix. So in simple words, this is a matrix that assigns a positive match score, a negative mismatch score, and a negative mismatch penalty. So according to what you would like to highlight in the sequence alignment, it is possible to assign different gap penalties for opening a gap and another one for extending a gap. So, for example, if we take a -10 score for gap opening and a -2 score for gap extension, then the number of gaps in the alignment is usually reduced, in the final alignment, I mean. So, the residues and the gaps are kept together, and this is biologically more relevant. So, if you are willing to have more biological relevance, you would use a higher scoring for opening a gap. And these are basically the options that you can use through the different websites when you are performing alignments. You can play with that scores. And there are different algorithms for global and local alignments. I'm just highlighting 2 of them here that are very famous, which is the Needleman and Wunsch algorithm for global and Smith-Waterman algorithm for local. So these are basically the 2 methods. So in the Needleman and Wunsch algorithms, what we do is just to align protein or sequences or nucleotide sequences, and I told you I will be talking only about DNA, so this is an example of aligning 2 sequences on the matrix. So the first sequence is here, the second sequence is here, and you would assign a different score to each position, and then you backtrack the alignment in order to take into account these the events that would have occurred that would help you to retrieve the most, what we call parsimonious, or the most likely events that have resulted in the sequences being aligned this way. So the